After studying this module, you shall be able to know about the nature and types of chemical evidences in forensic science, and you will also know the relevance of such evidences and the forensic analysis of those chemical evidences. Certain processes are more important to the society than solving crimes, both in terms of protecting the public from criminals and also to protect the innocent from undeserved punishment. Forensic science is broadly defined as the application of science to solve criminal and civil lawsuits. Very often, the strength of a prosecution rests on the capability of law enforcement personnel to hook up the accused with victim by comparing physical evidences collected from the crime scene or victim with trace or other evidences found from the suspected persons of the crime by analyzing them in the lab. Chemistry is also known the science of composition, structure, properties and reaction of matters. Chemistry has been described as one of the basic sciences because many other sciences arise from its principle. A major focus of the modern forensic science crime laboratory is to determine the chemistry of evidences received by applying the principles and laws of chemistry. The application of chemistry is to analyze the forensic samples recovered from the crime scene or victim is a major activity of forensic chemistry. Several forensic authorities have developed prototypes for forensic evidence. Forensic chemistry is one of the most important branches of forensic science which deals with the poisons, chemicals and the collections, preservation and analysis to determine their origin, toxic effects, quality and quantity for the purpose of justice. There are many facets of forensics such as drug analysis, toxicology, trace evidence analysis and arson evidence analysis where forensic chemistry plays a very important role. Analysis of forensic physical evidence such as powders, plant materials and arson debris are usually covered under forensic chemistry, while in forensic toxicology the analytical work deals with drugs, poisons and their biological byproducts in blood and bodily fluids. In simple words, both forensic chemists and toxicologists work with drugs and poisons, but both of them have to analyze different samples of evidence. The field of chemical analysis is the most diverse as compared to other disciplines of forensic science. Chemists have to analyze diverse range of chemical evidences that are encountered in different type of cases. The chemical evidence may vary from one case to another and also from a simple entity to complex mixture. Chemists analyze besides drugs, the paint samples, remnant residues of explosives, fire debris, gunshot residues, fibers, presence of radioactive substances in case of nuclear weapons, toxic chemicals in case of chemical weapons, and biological toxins in case of biological weapons. Forensic chemists can also be of great help in cases where specialized analyses are required such as environmental pollution to test the compound and quantity if required and trace their origin. A trained forensic chemist can determine its composition along with nature of material and predict its source as well. Modern chemistry employs a wide range of analytical techniques along with traditional methods of analysis. The report is presented in the court of law as expert testimony. Now, we will study chemical evidences are related with forensic chemistry. in this controlled substances or the drug of abuse. Controlled substances are chemicals legally recognized potential for abuse. They include street drugs such as heroin or ecstasy and prescription drugs such as oxycodone. Controlled substances can be classified on the basis of effect they produce, like stimulants or known as uppers, depressant also known as downers, hallucinogens and dissociative drugs, narcotics and opioids, and last other drugs of abuse like inhalants. Forensic drug analysis deals with the identification of illicit or the illegal drugs. Detecting the identifying controlled substances is a critical step in law enforcement fight against drug related crimes and violence. Now, licit and illicit liquors. The liquors are sold in the market in various brands and covered under Excise Act. The possession, sale, transportation of liquor is allowed only as per rules and regulations of excise and prohibition. Many times these liquors are being smuggled from one state to another. Illegal possession transported without proper valid documents. These samples are seized by the police and submitted to the forensic science laboratories for their examination. The liquor is examined in laboratory for two purposes. Firstly, for excise purpose, where mainly the presence of alcohol plays an important role, and accordingly the examination of liquor samples for the qualitative and quantitative is the main purpose of the investigation. Secondly, the liquor is examined for quality control, duplicate samples which are being sold in the market, in which the examination is carried out for the parameters also apart from alcohol contents. Now, the petroleum products. Usually, the examination of adulteration in petroleum products remains the area of forensic concern. Adulteration means illegal or unauthorized introduction of any foreign substance into standard petroleum product such as motor spirit and high spirit, etc. Petroleum products most frequently referred to any forensic science laboratory for examinations are petrol, kerosene, white and PDS, diesel also known as LGO and HSD, ATF kerosene, lubricating oil, furnace oil, waste oil, etc. These samples are forwarded to FSL for their purity, detection and adulteration if any, or under the Essential Commodities Act or under Section 420 IPC and also under Petroleum Act, unlawful possession, breach of contract, privilege, etc. The analysis of these petroleum products is a tough task, as these are complex mixtures of hydrocarbons obtained by fractional distillation of crude oil of variable nature. Now, fire and accelerants. The identification of fire accelerants, especially petroleum products such as kerosene or gasoline, is of great significance for determining the cause of a fire. Arson debris is collected and packed in tight containers due to volatile nature of compounds to be analyzed. Charcoal strips may also be used for collecting these volatile evidences. The chemicals from the debris are absorbed onto the strip and later dissolved in a suitable solvent before analysis. The use of gas chromatography mass spectrometer identifies the type of fire accelerator used and also determines the hydrocarbon content. Now, the detective dyes used in trap cases. Law enforcement agencies arrange a trap for illegal transaction or the bribe with the help of the complainant, where the accused is given currency notes on which chemical like phenolphthalein or anthracene powder has been applied. Nowadays, phenolphthalein is being used in most of the trap cases. If the accused touches the note then the part of the chemical which may be in traces is transformed on his hand or fingers. If he keeps these currency notes in his pocket, bag, briefcase or file etc., this chemical is also transferred to these objects according to the Locard's principle of exchange. In case of phenolphthalein, the object like hand, bag, pocket etc. are washed with a colorless solution of sodium carbonate or sometimes with lime water, which becomes immediately pink, confirming the touching of currency notes transferred of phenolphthalein to the subject. These washings are being collected and sent to the forensic laboratories along with other relevant articles to establish the presence of phenolphthalein, which can be considered as vital evidence in the court. The pink color of this solution persists for some days or months depending on the quantity of phenolphthalein and strength of the alkali solution. Now, the explosives. Explosives is a substance or mixture of substances capable of producing an explosion. This is a highly rapid oxidation reaction that releases a large quantity of energy and large volume of hot gases. Explosives usually involve terrorist attacks. Forensic experts are called to the scene of an explosion to determine if an explosive was used. It is also very important to determine what kind of explosive was used and how it was used. Therefore, knowledge of explosives and their characteristics is paramount for a forensic scientist involved in such criminal investigations. Some examples of explosives are RDX, TNT and PETN, etc. Now, the introduction to forensic toxicology and related chemical evidences. Forensic toxicology covers many scientific areas including analytical chemistry, pharmacology, biochemistry, physiology and pathology. In India, the forensic toxicologist is mainly concerned with postmortem toxicology, where a forensic toxicologist deals with qualitative and quantitative analysis of drugs or poisons, including their metabolites from the biological specimens, especially viscera, for medical legal purposes. The exhibit also include body fluids such as blood, urine and gastric lavage, etc. Forensic toxicologists can also encounter blood samples of live subjects for the presence of alcohol in driving under the influence DUI cases. Now, the principles of forensic toxicology. The objective of the forensic toxicology is to attempt to provide answers to questions that may arise during chemical investigations or in subsequent court proceedings. Following issues are to be resolved. Like 1. Was the person being poisoned? If yes, then what type of poison was used? Identify. Now, how was the poison administered into the body? The kind of mode. Then, was it sufficient to cause death? The lethal amount. Now, forensic toxicology involves following types of chemical evidences. The first one is plant poisons. Plant poisons generally belong to a chemical class called alkaloids, the same group that contains caffeine. There are large variety of poison plants reported worldwide like Dhatura, Calotropis, Oleander, Ricinus and Argemone are mostly encountered in Indian conditions. They can have different modes of action on different parts of human and livestock. The second is metallic poisons. Accidental poisoning due to heavy metal still occurs from materials in the environment. As with many heavy metal poisons, its effect tend to be more severe in children than in adults. Mercury is another environmental poison, particularly when it is in the form of methyl mercury or dimethyl mercury. The third one is gaseous poisons. Poisons, toxic gases are responsible for a large number of deaths each year. One culprit is carbon monoxide, produced as a byproduct of combustion. Anytime there is a fire or open flame, significant amount of carbon monoxide can be produced. Cars are based on internal combustion, so car exhaust contains carbon monoxide. As a result, vehicle exhaust is sufficiently poison to kill human accidentally. Accidental carbon monoxide poisoning occurs when heaters that use flames malfunction or are brought into a home. Another combustion product, HCN, is a very toxic substance that can also be formed when synthetic materials such as plastic or fabric burns. Now, forensic analysis of chemical evidences. After a police officer or investigator has collected evidence at a crime scene, some evidence may be brought to the crime scene lab for forensic chemist to analyze. The chemists follow a specific process based on the scientific methods for analyzing evidence. Samples collected from a crime scene and brought to the lab for analysis are called quotient samples because the identities and origins of those samples are unknown. In order to draw conclusions about the identity or origins of quotient samples, the forensic chemist will need known samples as a reference. Forensic analysis may be performed to identify a quotient sample or compare a quotient sample to a known sample for the purpose of determining the source or origin of the sample. The result of such comparisons can link a quotient sample and several known samples either to a class of samples with several possible origins like classifications or to a single origin individualization. Now, identification. When a quotient sample is submitted to a crime laboratory for analysis, the first task is identification. One of the key methods for identifying the type of evidence at a crime scene or in a laboratory is the use of chemical reactions. Now, physical and chemical properties. Physical properties can be described or displayed without investigating chemical changes. Chemical properties are properties of a substance that can be described through a chemical change only. Chemical changes require a chemical reaction to occur between reactants generating new products. Now, presumptive analysis. Observation of the chemical and physical properties that are not unique enough by themselves for identification but that provide enough information to narrow the search. Presumptive analyses are usually quick and inexpensive to perform. Now, the color test, also called spot test or presumptive drug test, which are used to probe quotient drugs samples for their chemical properties. Although these tests are fairly accurate and not the final test used as absolute proof evidence in court. These tests are less expensive than the final test performed in the lab. Most color tests are extremely simple and only require the addition of a prepared solution followed by a visual observation. Now, the microcrystalline test. A technique considerably more specific than color test is the microcrystalline test. Under microscopic examination, the size and shape of the crystals are characteristics of this specific chemical. These tests are cheap, rapid and often do not require isolation of a chemical from its diluents. Now, the next is thin layer chromatography (TLC). Chromatography itself covers a large range of applications in forensic examination. Thin layer chromatography incorporates TLC plates either manually made or ready-made or pre-coated. Sample is spotted on the plate and plates are made to run in a suitable solvent system. Confirmatory analysis. When presumptive analyses are negative, they exclude potential drug candidates. When they are positive, they direct the forensic scientist towards variable confirmatory analysis. Spectrophotometry is a measure of how a substance reacts to UV and IR light. A spectrophotometer emits different frequencies of electromagnetic waves in UV and IR spectra and simultaneously measures the reflections given off by a sample. The selective absorption of light by different chemicals in the UV regions of the electromagnetic spectrum provides a valuable technique for identification. Now, mass spectrometry. The technique of gas chromatography is particularly suited for analyzing drugs, explosives, and poisons since it can readily separate a chemical from other substances present. Now, next is comparative analysis. Many forensic analyses end with identification, for example, identifying an unknown substance as a drug, explosive, or accelerant used in arson. But some proceeds on to comparison. The purpose of a comparative analysis is to link a quotient sample and a known sample to a common origin. The origin may be broad, resulting in a classification, or exclusive, resulting in individualization. Now, the next is chain of custody. A chain of custody is established and documented as the evidence is sent to a forensic laboratory. The most common aspect of all forensic chemistry is maintaining the chain of custody. Chain of custody refers to the time course in which evidence was handled and includes every person who handled the evidence. It is imperative that evidence be handled by the minimum number of people needed to complete the forensic analysis. An ideal chain of custody is one that involves two individuals, one person who collects and delivers the evidence, and one person who analyzes the evidence. All persons in the chain of custody must be prepared to testify in court to validate the integrity of the evidence. If the chain of custody is broken in any way, the evidence will be excluded from court testimony, which often results in the dropping of criminal charges against the defendants. Forensic science is broadly defined as the application of science to solve criminals and civil lawsuits. A major focus of the modern forensic science crime laboratory is to determine the chemistry of evidences received by applying the principles and laws of chemistry. Chemists have to analyze diverse range of chemical evidences that are encountered in different types of cases. Controlled substances are chemicals legally recognized potential for abuse. The liquor is examined in laboratory for two purposes. Firstly, for excise purpose, where mainly the presence of alcohol plays an important role, and accordingly the examination of liquor samples for the quantitative and qualitative is the main purpose for the investigation. Adulteration means illegal or unauthorized introduction of any foreign substance into standard petroleum products such as motor spirit and high speed, etc. Forensic toxicology covers many scientific areas including analytical chemistry, pharmacology, biochemistry, physiology, and pathology. Forensic analysis may be performed to identify a quotient sample or compare a quotient sample to a known sample for the purpose of determining the source or origin of the sample.